Now, I'm sure you were a pro at what you just did, so let's try to make things a little more complicated. Now, the nice thing in genetics is if we want to make things complicated, all we have to do is add one more gene and boom, we've increased our complication factor exponentially here. So we'll still take it kind of slow. We're just going to try to do 2 genes at once now instead of 1. Let's try to look at the trait of lactose intolerance and dwarfism at the same time. Now, the lactase gene is on chromosome 2, and the FGFR3 gene, which controls the trait of achondroplasia, which causes dwarfism, is on chromosome 4. And let's say we have an individual who's heterozygous for both conditions. They're heterozygous for lactase persistence, so they are lactose tolerant, and they're also heterozygous for the dominant allele that causes dwarfism, which means that they're also going to be a little person. Now, to help us know which plus and minus symbol goes with which trait, I'm going to put an L next to the alleles for lactose intolerance and a D next to the alleles for dwarfism. Now, instead of trying to figure out all the probabilities of what the next generation would be, let's just focus in on how this one person who's heterozygous for both of these traits is actually going to make their gametes. And what are the possible allelic combinations for them to make their gametes? So after meiosis, how many different allelic combinations or genotypes can there possibly be for this individual? They're heterozygous for the lactase gene, and they're heterozygous for the gene causing dwarfism.